That's a load of science.
Hello, everyone. Welcome to another episode of That's a Lot of Science, the podcast for science lovers and everything about science. Today, we're going to talk about a very important topic, a breakthrough discovery that is changing the worlds of medicine and beyond, called CRISPR. And we're doing that with our guest today, Ruben Vasquez Uribe, CRISPR expert, of course, and microbiologist. So, first of all, Ruben, thank you very much for being here with us today to talk about this very interesting topic. And as I always like to start with my guest, I leave the stage to you for a couple of minutes, so then you can talk about yourself, talk about what you are doing, what are your passion, what are you doing now. So please.
Yeah, well, hello and thank you for inviting me. Yeah, as you mentioned, my name is Ruben, and I do have some experience in CRISPR. In general, I'm very passionate about microbes, all type of applications, how we can harness them to apply them for different things. So fermented food, the impact in the gut microbiome, and even how to wire them in different ways to create new functions. Um, so I have this background in synthetic biology and in microbiology, and yeah, I currently work on how to use engineered microbes uh, to improve health through the gut microbiome.
Okay, very, very interesting. Okay, thank you. And you already mentioned the, engineering of microbes, and CRISPR is one of the methods that is used to do that. I mean, but it is sometimes referred as a single thing, but it is actually a, a lot of techniques, a lot of methods combined in CRISPR and how they are performed. And how are they doing? I mean, how do you do it in the lab? How do you perform this in the lab? And what are the scope of this experiment and this technique?
Yeah. So I think that is one of the things why CRISPR have become so powerful and popular is because it's relatively easy to implement in almost every laboratory setting. So I'm going to talk specifically about microbes, but so to use CRISPR in a bacteria, all you need is a vector that expresses the the nuclease, the effector of the CRISPR system, like Cas9, and then a very short piece of RNA that will guide the nuclease to cut in a specific site. These two things can be encoded even on the same plasmid, and nowadays, I mean, uh, cloning these or sometimes even just ordering some of these vectors ready to go, it's relatively straightforward. And I mean, when it all started, like back in almost a decade ago, it was mainly implemented in E. coli, but over the years, I mean, you see more and more publications coming out implementing it literally across very different microorganisms, and it keeps working. It just works. So it's relatively easy in that way to implement.
And the fact that works also allows you, I mean, the good thing is that it allows you to do a lot of things, right? I mean, it can be applied to many things, and it's not just, of course, you mentioned microbes, medicine. It is important that I think they keep it easy in the lab, but also it has a very wide scope, no? I mean,
Yeah. I mean, it has, I mean, besides microbes, it has also been implemented in yeast and in many other multicellular organisms, including humans. So yeah, I mean, we just see that that is that powerful. I mean, in a way, I mean, it's one of the most revolutionary technologies in science. I mean, it's a before and after CRISPR. Uh,
yeah, there is a before and after CRISPR era. And we are definitely now in the after CRISPR era. And you say this started like a decade ago. And we know in 2020, the inventors of this gene editing technique were awarded the Nobel Prize. What happened in this decade? I mean, in terms of advancement, in terms of changing the technique, or like, I don't know, broadening the scope,
Yeah.
what is happening?
So I think in the last decade, what it has happened is that we discovered that there is a lot more CRISPR systems out there in nature than we thought of. I mean, the inventors that received the Nobel Prize, they use this CRISPR system that uses Cas9, and just to differentiate between these two things, so the CRISPR part is just the RNAs that guide the nuclease, and the nuclease are Cas proteins. So the Cas9, it's the actual effector, is the thing that actually cuts the DNA. And in the last decade, we have discovered that Cas9 is just one of so many others, many other different types with different functions. Some cut RNA instead of DNA, some cut both RNA and DNA. We can also modify these effectors so that instead of cutting, they can silence genes. So we remove the catalytic effect of the enzymes, and then we make them only bind to specific sites. We have discovered that they have a lot more functions than just cutting the DNA. So I think that's what has mainly happened in the science space for CRISPR. And of course, that we have applied it to many other organisms, and also how we translate it into therapeutic.
Yeah. That's a very, very strong topic then. But you mentioned like different Cas types, and this is like something like still in the technical area, in the scientific area. I mean, how do you pick one? Depends on the organism, depends on what type of modification or editing you want to do, or is just like, whatever. Of course, it's not whatever works, but
Yeah.
it changes on the type of gene that you want to insert. How do you make the decision of which model and which strategy to use?
Yeah, it depends on your application.
Mm, mhm,
Uh, because Cas9 has been so studied, it can be applied in so many organisms, but it's actually a relatively large enzyme. There are other effectors that are much smaller, so it's easier to pack them, to deliver them through vectors. It also depends, for example, if what you want to do is to cut RNA, then you wouldn't use the Cas9 or the other ones. You would use the Cas13, which is the other CRISPR system that can cut RNA.
okay. So it
So it
depends.
depends on
Yeah,
the
like,
application.
it depends on
Yeah.
the application. And what would you say? Like, I mean, of course, it's very important that this system cuts in a very specific loci of the gene. No, they don't cut anywhere. They're very accurate. And is that something that is in, it is recognized as a challenge? I know this has been a challenge, not the accuracy of the method, making sure that it cuts in the right place where to insert your gene or whatever. But how is this being solved, if it is, or is still a problem in using this technique?
I think it's less of a problem because we have engineered better versions of these effectors that are more accurate and less prone to make errors. Also, the way that we pick the targets matter, because of course there could be some regions of the DNA that maybe are more unique. And CRISPR systems are really good at recognizing specific sequences, but because they are the immune system of bacteria, they also allow for some degree of dissimilarity. So some of these off targets can also come from this wiggle room that the CRISPR systems have to recognize as a perfect match. So
Mhm.
it doesn't have to be a perfect match to have an effect. Yeah.
Yeah, that makes sense. And as you mentioned, yeah, CRISPR comes from the immune system of bacteria. So actually, I mean, we engineered a process that was innate in the nature of bacteria, and make them work for our application. But how was that discovered? I mean, how they came up with this idea starting from this, you know, this system, this immune system that bacteria has already.
Yeah, I mean, science being science, it actually just started, as observations. People started to sequence. I think it was Francisco Mujica in Spain. They started to sequence pieces of DNA from bacteria, and they started to look these very odd, repetitive regions in the genomes of bacteria. And that was, I think, in the 90s. So it took a while until people pieced together that those repetitive regions actually match pieces of foreign DNA elements, plasmids, and bacteriophages, which are usually threats to, or they could be threats to certain bacteria. And it took a while until they figured out that those pieces of DNA, they are transcribed into guide RNAs that together forming a complex with the effector can guide uh, um, yeah, the effectors to detect and cut uh, those type of elements.
Okay, okay.
So yeah, from Francisco Mojica, I mean, you go all the way to observations that it's a complex of the Cas effector and the guide RNA, and then it was until much later when Emmanuel Charpentier and Jennifer Doudna actually, they are the ones that received the Nobel Prize. It was much later when they actually were like, okay, so actually Cas9 can form a complex with these. And at this point, they already knew it was immune system, by the way. At this point, they already knew that.
Hmm.
Like they received a Nobel Prize because they showed that it could be reprogrammed, that you could take the factor and guide it to a place where you want it to cut specifically. And they show that in bacteria and later in human cells. And I mean, that's why they were awarded that. But it was actually much earlier that other people started to piece together the pieces that it was an immune system and that it was not only an immune system, but it was an adaptive immune system, that it could remember the encounter with a previous um, organism, and it would be specific. And this is very different from other immune systems that we knew from bacteria before, because we knew bacteria had some sort of innate immune system. So, for example, restriction enzymes. I mean, those. I mean, if you have been doing cloning for more than 20 years, you might know that back in the day, cloning happened with restriction enzymes. Nowadays, people don't use them so much anymore. But again, we use that immune system from the bacteria to repurpose it and cut and paste DNA. And in this case, I mean, people knew the existence of this, but this is an innate immune system. It's just there, it's just on, and it doesn't adapt to specific threats.
Yeah.
The difference from CRISPR system is that it would adapt to it, remember it, and be able to pass it also to the next generation of cells. So it's very sophisticated. I think the biology alone of the CRISPR systems is very interesting.
Yeah, no, absolutely, absolutely interesting. And as you said, it's not just the work of one. No, it's been like I mean, since decades ago that the observation, and that's how science work for everyone. And it's like people are like, observing a phenomenon or observing something, and then on that discovery, someone else will be able to build upon that, and then on top, on top, on top. Yeah, what is innovative, what you said, it's very interesting that this system is adaptive and can be transmitted into the germline, so it is then transmitted to the following cell. And
I mean,
yeah, go ahead.
if I can just add something to that, I think
Yeah.
the interesting part is that probably before 2010, people didn't pay that much attention to these presentations, People would go to conferences and yet someone else would talk about immune system of bacteria and something called CRISPR, whatever. I don't think that people gave that much attention to it at that period because we have known about these repeats for a while,
Mhm.
and people had a hint that they were immune systems and that it was possible to form a complex between a protein and an RNA to direct them towards specific cutting sites, but no one really paid attention to it. The people were just talking about it. It actually only took a conversation of Emmanuel, or at least the legend goes that it was a conversation in Hawaii during a
Mm-hmm.
conference between Emmanuel Charpentier and Jennifer Doudna, and then from relatively different fields, and then all of a sudden, boom, it sparked like, hey, this is actually very cool. Like we can actually, this might be really big. And then
That's a good thing. Yeah, how science works sometimes. Not two different minds getting together, finally making sure that they listen to each other, and bam, the
yeah,
new
but it
idea
also
is out.
makes you it makes you wonder like what other like discoveries might be hidden in a conference. I mean, we always
Just
only put a, you only we always only put attention to our topic or our field. But I
yeah.
mean, we might be missing on the next
Yeah,
CRISPR.
or yeah, you might be, I don't know, you might go to a conference, talk with someone, have a very interesting idea, but then you might not have time to develop it because you are like overwhelmed with your daily work, and you're like, okay, it's maybe a good idea, but let's keep it in the drawer. I mean, that's something that push forward in terms of collaboration, talking about people, networking, because you never know what it might come out of this. And we have seen with this how the interest sparked, and now there is maybe, I don't know. I mean, do you feel like there is a hype on that, or the hype is real on CRISP?
I mean, maybe it's because I lived very close to the to that period, but I think the hype is a little bit less now. I think, I mean, starting 2011 until a few years ago, it was what I would call the CRISPR craze. Everyone wanted a piece of it when they realized that they could edit DNA and that had a huge potential. So many legal battles started and patents and discoveries, and I mean that was really like a rush to try to discover another CRISPR that you could patent that is not related to the one from Doudna and Charpentier and Feng Sang and all of these things. Um, and yeah, I mean, I think there was so much hype during this
Yeah.
period, and like I think even Netflix had like a documentary about CRISPR, and it really made reevaluate like a lot of these things of like, okay, now we actually have a tool that is very powerful, that is easy to implement, that is easy to program. What are we allowed to do?
Mhm.
Yeah,
Yeah, yeah, that's true. There was I watched the documentary. I remember that.
yeah,
I
it's
remember
good.
that
It's
when it
good.
came out. Yeah, no, it was a good one, and it's important that they also, I think they had I think they had both inventor like Charpentier and Doudna. They were interviewed in the documentary as well. So I mean, at
Yeah,
least they had safe sources
yeah.
there. And
I
yeah, you are right. I mean, it's the same that I'm seeing with the AI at the moment. Now everyone wants to have the AI label, and there was this kind of things few years ago. Now everyone wants to have the CRISPR label on their product or on their process or whatever. So
Yeah,
no, it is kind of on the same line.
but
And
I think it tells
yeah,
you how impactful it was because it permeated into the general public. I mean, I watched this documentary with my mom, and my mom is not a scientist. I mean, I think she would do a really good scientist, but she's not. She has studied civil engineering. And I remember watching this documentary, and at the end, my mom was like, "We should just CRISPR everyone." And then I was like, "I don't think that's the point."
that's not exactly that's another thing, well, how these is then used, because sometimes I mean, the general media tends to kind of from one way build the hype, but this would lead to a little bit of misinformation. And people were thinking about that, like, why we don't just treat everyone that has a genetic disease, or why we don't design our babies or stuff like this. And this is a big concern for now, being using these into humans. And I mean, of course, at the moment, one of the approaches that is being used for using CRISPR in therapies for human, it's more for those genetic disease like sickle cell disease, no, where you have one specific mutation that you can engineer. And I know there is, I mean, there is like an approved genetic treatment for that. There is a I mean, a lot of development, like drug in development, like biological drugs in development right now. But still, like, there are still concerns, no, on using this, in
Yeah.
particular for more complex disease, where we're talking about many genetic mutations that might happen, like in cancer or Alzheimer.
We also don't know what could be side effects of doing this genetic thing, um, or long-term consequences. I think, I mean, my personal opinion is that it's a very powerful technology, but we shouldn't be naive ab- uh, of and think that we fully understand
Yeah,
what's happening at the molecular level. And I
yeah.
think that we need a lot more research before we start to CRISPR babies. I understand that if a person living with sickle cell anemia has the chance to improve their life, that it's not up to me to decide if they will get it or not. Um, but I do think that as a scientific community, we need to be a lot more cautious of rushing this. Um, yeah, that being said, we can also not be naive to think that other countries are not thinking the same way. I mean, you
Mhm.
might have the European Union regulating it and having all of these ethical discussions of when to apply it and when not, and so on. But is everyone thinking the same way? Is everyone uh, trying to take the same steps to make it safe and so on? I mean, we live in a relatively globalized world. So if someone wants a CRISPR baby and they're in a part of the world they can get it, they will go there and they will get it
Yeah.
if they have the resources.
That's
Uh,
that's a strong point, because then the resources will make the difference, because of course, like, it is a, as you say, the relatively simple method and whatever, but it is expensive, and this will even increase the gap from, I mean, between those that have the resources to access this sort of therapies and this sort of technology, and people that who don't. And
yeah.
it's
And
important that, first of all, we have a global regulation, but also making sure that uh, the scientific application of this technique goes together with regulation and ethical application worldwide, because otherwise, yeah, you might have those discrepancies in people that would actually need but cannot access it, and people that would probably be fine even without it, but just because they want a designer baby, they would access it anyway, limiting the access to the other one. So that's
and you
something.
know, it's a slippery slope
Mhm.
from treating a disease to treating something that some people would consider it a disease and some people would consider it maybe not.
Yeah,
And then the next step, as you said, designer babies. I mean, I don't think I've been completely uh, extreme to think that people would want that. I mean, to be able to predict certain features of their
yeah,
children. I think probably there are people out there that if they have the resources
they
would pay
would.
for that.
Sure.
And I think that's yeah, that has a lot more implications than just
It's very,
curing
yeah,
a
no,
disease.
exactly.
Yeah.
It's very frightening where this can go, and it is important that the scientific community in that support the type, in my opinion, what type of this kind of approach and mentality, because again, as you said, there are people that might want to access it and potentially have the power and the resources to access it, and we need to make sure that what we do is always ethical, but at the same time, we cannot limit the scientific advancement, because,
Yes.
as you said, in the last 10 years already, this method blown in many different applications, and we don't know what would happen in other 10 years. And again, what will be the regulation in 10 years? What will be deciding and advocating for this in 10 years?
I think
We,
for me, that's the key message that we shouldn't limit innovation. I think we should guide it so that you know we take the right steps to make sure that it's safe, to make sure that uh, it will help people. And Europe and the United Kingdom, they are really good at making sure that, but maybe too good. I
Yeah.
think we also need to make sure that we allow a space for innovation.
Yeah, exactly. And make sure that the regulatory framework that we put to this would be exactly, it would be not a cage, but at the same time, not just something that can be used without responsible use from everyone. I mean, from those that have access to it. So these are, I mean, as you say, these are also CRISPR is interesting because it is a germline mutation. So it is something that once you allow someone to access these features, can be also transmitted to his prole and so on and so forth. So we are kind of, messing up, I would say, with what humans are, and we want to make sure that we don't mess up. We do it in the right way. And I mean, clearly a very strong message there, making sure that we still advance because many great things can be done with this
Yeah.
technique. And we already mentioned the therapies that are now in the market and clinical trials that are now being conducted. But at the same time, ethical consideration never to be forget. And okay, changing a little bit of topic. In your daily job, no, you focus on application of this technique mainly in microbes. What is the interest? I mean, what is the aim of that?
Yeah.
What do you use it for, and how do you use it for?
Yeah. So, I mean, that's another thing I'm very passionate about, the gut microbiome. I think this concept that, uh, and I know that you had a colleague of mine in one of your previous
Yeah.
podcasts, Emily, um, and the reason why we're so interested in this topic is because it's this idea that we have a part of us that makes us us,
Yeah.
but it's not human cells. It's actually microbes, and they change through life and through our diet, and they have a huge impact in our health. I think probably, I mean, the topic of microbiome is not new. I mean, we know about its existence like for more, I don't know, I would dare to say like more than 50 years probably. We know there are bacteria living there. We know that. And we know it's very diverse, but it was only because of sequencing methods that we were allowed to see the diversity and the connection to certain diseases. And in the past 20 years, the field has really developed.
Mhm.
And now, I mean, there are some links to certain diseases like inflammatory bowel disease or colorectal cancer, where the link is pretty strong with the gut microbiome. I mean, for others, I think that we keep researching to try to understand how they are connected because we can see a link, but we still don't know the mode of action, how specifically they are linked, but we know there is an overall effect on health. The way I look at it is almost as if the gut microbiome it's an extra organ.
Yeah.
Is people describe it as a second brain or something like that. I mean, I just see it as an endocrine organ. It just sends signals to your body, to your immune system. So like any other organ, if you lose it or if your organ is sick, you
Mhm.
will have consequences in the rest of your body because everything is interconnected. So the reason why I study it is because it plays such an important role, but we don't have therapies to treat it nowadays. I mean, we know if you give antibiotics to a person, it will clear out the
The
gut
flora, the
microbiome.
gut.
Yeah,
Yeah.
but what happens if you only want to remove a few species? What happens if you are like, okay, I know this particular species that I have in the gut microbiome are promoting a more dysbiotic environment? Um, and if you would like to specifically remove those, you would like to have a type of antimicrobial that can do that.
Mm.
Nowadays, antibiotics, they are basically like a nuclear bomb. I mean, they kill everything
Yes.
around. Um, but during my PhD, actually, I worked a lot in a technology. Um, to develop what is called CRISPR antimicrobials. So you take the CRISPR system, you pack them into a vector. It can be a phage, it can be a conjugative element, and you deliver it to the target bacteria. Then the target bacteria starts to express this vector, but this Cas9 and the CRISPR system, they target the chromosome of the bacteria. So they actually destroy the chromosome and they kill it. But because it's targeting specific sequences, it can actually differentiate even at the strain level, because you can have two species next to each other, but only one of them maybe, let's say, has a virulence factor or an antibiotic resistance gene. And then you can direct your antimicrobial to only kill that one. Um, and that's just the beginning, right? Like killing, that's just one function, but you could also replace genes or add genes or remove genes. So you can edit the function of the gut microbiome. And we know CRISPR works really well in microbes. First of all, it is from microbes. It is their immune system. It works really, really well. So it's this idea that maybe we can alter the function of the gut microbiome or the composition so that we can have impact on health.
Yeah. Yeah, no, interesting. Yeah, I mean, first of all, yeah, what you said about the antimicrobial, very, very interesting, because of course, again, we are experiencing, strains of bacteria super resistant against various, antibiotics. Some people might get infectious diseases that cannot be treated with antibiotics. This can be a way, to specifically target those, but also not just the microbiome one, but in general, when you have an infection, as well, something like when you have infection from a bacteria that would not respond well to antibiotics, this can probably be a different approach towards this kind of disease. But also, as you said, make the engineer your gut microbiome to produce a certain metabolite, or to be able to, I don't know, metabolize faster some food. And we know this can be useful for, I don't know, weight loss or for other applications. We know how the importance of the microbiome is in our regulation, and not just the digestive system, but all the other systems. You mentioned the immune system. We talk about the brain as well, not the brain gut axis, but also the brain microbiome itself. No, there are some now there is this so-called psychobiotic, no, this sort of probiotics, or like microbes that have a direct impact on the brain itself and the brain functioning. So is there sort of a, interest in the health, of course, for the microbiome, but do you see that instead of the classic probiotic, there is an interest in having engineered microbes for specific disease? I mean, do you see that in your experience? I mean, you mentioned IBD, but something beyond the gut health? Do you see that?
I, I think the more we understand the gut microbiome, the more we see that there are specific targets. So one of them is the modulation of the immune system. The gut microbiome actually has a huge effect on that. Um, so I mean, I do see, and our immune system is what hold us from falling into different diseases. So I do see the potential for therapeutics. But I think um, uh, it's not only limited to drugs, even just to improve or enhance the features of already existing probiotics. I mean, probiotics, it's a huge market. People really like them. People really want to use them to improve their health. A lot of these people are not even sick. Some of
No.
them, they just want to prevent being sick and being healthier. But actually, the term probiotic is one little term for a huge umbrella of different bacteria with different functions. Our microbiomes are so diverse, and study after study after study, what they encounter is that people respond differently to probiotics. And the ones that respond, maybe it's a very small signal. they don't really get the effect that they are searching for. So, I mean, why not then take some of these probiotics and enhance some of their features with CRISPR, with other tools, to have more predictable effects. Something that you can say like, hey, you want to stay healthy, you want to have better digestion or less inflammation or so on. Take this, and then the person is guaranteed that it will actually do the function that it's supposed to do. You would not be selling a snake oil. You would actually be selling something that is validated and that is for healthy people. It's not only to treat diseases. So I do see the potential as therapeutic. I think that's very obvious to me, but I see
Mhm.
also that potential for an alternative for people to also stay healthier.
Yeah, that's true. And also, as you said, no, every people responds differently. So, I mean, our microbiome is like a fingerprint. No, I mean, there are no two people that have the same microbiome, and this leads then to the personalized medicine that now it's like on the rise. So it's not just one medicine fit for all, but
Yeah, that's a
one
good
one
point.
person, one medicine. So
Yeah.
then, I mean, the microbiome is probably the best example for that because they are so unique in their composition from human to human that really opens up possibility for personalized medicine or for like exactly personalized, not necessarily therapeutics, but something to improve the health or to improve the quality of life of
Yeah.
people that want to use that.
And you know what? I think why it's so interesting for CRISPR is because right now we don't have therapies for the gut microbiome. I mean, we know probably eating better works, a lot of fiber,
Yeah,
probably fruits, vegetables, nuts, all of that. Probably we should be eating it anyway.
yeah,
But we know that already for like hundreds of years. And yet again, I mean, people also prefer to have a pill to treat
yeah.
things. So, yeah,
Yeah, also a change of mentality, like thinking about not having a disease, but like, do all the steps required to be healthy without the need I mean, of being sick in order to treat yourself right. I mean, we know, like, everyone prefer to, do the pill at the end, but maybe it's also a change of mentality and make sure that people follow those steps in order not to need the pill. But that
yeah,
maybe is asking a bit too much. The
yeah.
people, I don't know. But anyway, I mean, but beyond the health, do you see, like, I mean, of course, engineer a microbe can lead to other applications. Like, I mean, we can make them produce something for us that can be used for something else. No,
Yeah.
I mean, do you see that going on? No.
For microbes specifically, or
Yeah, yeah. I mean, whatever you might have encountered, if there is an example of something that you think it's interesting of an
could you
application that.
repeat the question? Sorry.
So instead of having an engineered microbes for specific health condition, I don't know, for any other industrial application or like any other um, sector beyond health that can be interesting in having engineer microbes or engineer. I mean, microbes sometimes are seen as a small factories, no, because they
Yeah,
produce so many things. So I mean, engineer like a microbes for other application that you might have
yeah.
experience or you have some example.
I mean, first of all, I think they have a huge uh, potential effect in for environmental remediation. Some of the first examples decades ago, actually of engineered microbes, it was to degrade toxic components in soil or in water. So I think there is also a huge potential there. Again, I think the problem with that is also that we are only starting to extend that technology. And again, the same message. I don't think that should stop it from being implemented, but I think we should take the necessary steps to make sure it's safe.
Mhm.
But again, I'm always pro testing some of these things. So there is environmental part. I think there is also a potential impact on how we produce food.
Mhm.
So for example, the same way that we have a microbiome, Plants also have a microbiome, and depending on the microbiome, their yield might be different, or they might be prone or less prone to certain diseases. So the idea that you can also, yeah, help in those processes, and then beyond that, also in how we affect the texture of different products with food fermentation. I mentioned in the beginning that one of my biggest passions is for fermented foods, because it combines two of the things that I like the most, which is microbes and food. Uh, and I think that, I mean, if you take some of the most delicious foods out there, you have cheese, you have wine, you have bread, everything that is good in life, it's produced by microbes. So why are we not using them more? I mean, and this again is where CRISPR can come in, because we already have the fermentation processes, but can we bring CRISPR into the picture so that maybe we can add health features, or we can add flavors, or we can add textures to already the fermented processes? So that we can create, for example, foods from plant-based materials, but that they give all of the nutrition and flavor
Yes.
that people expect. Yeah. So
Yeah, yeah,
I,
yeah.
I
From
think
yeah, exactly. Yeah.
beyond health, for sure, there are a lot of applications for CRISPR and engineered microbes.
Yeah, because it opened it paves the way of having I mean that's the thing, no, having microbes that do what you want. I mean that you design the microbes to do what you need, and then of course you can apply it to many. Food is definitely one of the topics. First of all, because it links to many other things. It links to health. It links to environment. Because of course that's another thing. No, we ensure that our crop is healthy, that we don't consume too much of land. We don't, I mean, we know the farming is one of the most polluting industries out there. We are an increasing population of humans, meaning an increasing need for food for everyone, affordable food, and
Yeah,
yeah,
but not
absolutely.
every type of food, because I think that's what we didn't get so well in the past 20, 30 years. I mean, we mass produce food, but a lot of this food is not good for your gut microbiome, it's not good for your health, or not good enough. And we can make it better now that we understand that. And it's also not good for the environment how we produce it. So maybe we can find ways of producing more sustainable, delicious food that is also healthy. Um,
How do you
yeah.
think this will be seen from the general population? Like, I mean, we know, like already with the
Mhm.
GMO has been kind of, this is, I know it can be a tricky question for like, I'm just looking at your opinion, but I know that already answers some of the questions. But that's the thing, no? Imagine how many crop we could save using GMO, how many how much we could increase the yield of the
Yeah.
food. But still, there is some concern and some, I wouldn't say fear, but doubts about using that. And is this being accepted a little bit more? Is this, at least in the food industry,
Yeah.
we already touched upon the health and the human therapies, but
Yeah.
in this,
We live in Europe, and it's one of the toughest places for GMOs. But I think that's part also because in general, the public opinion on GMOs is not very positive. Um, and I mean, I often do this exercise with some friends, where I just ask them out of curiosity, like non-scientist friends,
yeah.
what do they think about GMO? Because I'm actually curious. If you ask most scientists, I think most scientists, because they understand GMO, they would probably be, or there would be a tendency to be positive about it, I think, between scientists. But I do think that the negative public opinion comes from not understanding enough of the consequences of GMOs. I had friends that they don't differentiate between organic and GMO, or they link something of pesticides and all these things to GMO. It's just that the general opinion of public is like, in general, we should do more natural things, and GMO is not natural. So why would we be doing that? But then you tell them like, hey, but wouldn't it be great that there are like plants that need less water, but they produce more? or that they are resistant to some pests, or that they are more nutritious. And a lot of them is like, yeah, that's a great idea, but I think it's a lack of understanding sometimes of the potential of CRISPR and of genetically modified organisms. And I think politicians in Europe or in the United States or in other countries, they are usually driven by the public opinion. So if the public opinion is negative for genetically modified organisms, they're not going to touch that. I think
Yeah.
it has to start with podcasts like yours that
Thanks.
are used to disseminate more disinformation, all the potential, all the cool things that we could do with genetically modified organisms, so that the public opinion also changes, so that the public opinion understands better
Yeah, yeah.
this potential, and hopefully that can influence also the direction of policies.
Oh, I mean,
Oh,
thank you.
yeah.
Thank you very much first, but I agree with you because it is, I mean, as scientists, it is also our, I wouldn't say duty, but it is on our shoulder also to teach on the, on the public opinion and try to disseminate as much
And
as we
we're
can
so
this
bad
kind of
about it.
notion. Yeah.
We are so bad about it. That's the thing. Like,
Yeah,
I
yeah.
mean, when we try to talk, I mean, we just don't make it approachable sometimes. I
Yeah,
think
yeah,
we
yes. I
could
mean,
do a lot of work on that.
that's another problem. I mean, the communication with people that are not in the scientific, I mean, in STEM in general, because I mean, if you have a scientific mind or something that is leaning towards scientific topics, you might be more, open to accept these kind of technologies and these kind of things. But at the same time, so it's very difficult from our end, and this is something that is our fault, maybe, like to communicate properly. And so we leave that to external and people that are not in science and people that might not have the correct knowledge to disseminate properly the topics and disseminate properly the potential, as you said. I mean, so yeah,
Yeah.
keep doing. I have to start doing this game with my friends as well. Then.
Well, I mean, I think for me it's just to see how I can communicate it better.
Yeah,
Because
yeah,
I was like, I mean, what questions do I need to answer? From where do I need to start if I talk to a general audience of people that went to university and that have maybe a bachelor's or master's degree? From which level I have to start to, or like no, not level, but like more like how can I change the way I explain some of these things, so that people are also attracted to listen more about it and that they can see the potential. I mean, because the thing is that we just focus so much on the science and publications and conferences and patents and startups and whatever, and which is very, very important, obviously. But then we don't communicate any of that to the general public, and the worst case scenario is that sometimes there is like a TV show or like Hollywood movie or whatever where they take the name CRISPR, and then they put it as like maybe something very negative or something
yeah,
very stupid just for their plot. It has
yeah,
no
exactly.
scientific basis, and then all of a sudden CRISPR becomes a completely different thing for people. I remember, I think a few years ago there was this movie called Rampage or something, where they do mention CRISPR, and is these huge monsters that invade like a city, and they are genetically engineered with CRISPR. And then I was just thinking, like, this is
No, 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 that's not good.
yeah.
That's exactly. We don't have good. I mean, these are not good publicity for us. No, we need to get a good publicist to send the message
Yes.
correctly, because they always depict the scientists as the bad guys, or like someone that is so crazy that cannot understand the consequences of what we do. But we are really, we are super aware of what we are doing and what are the consequences. That's why we are so cautious and we are so careful about using it. But yeah, you are right. I mean, it's important that we communicate this. And CRISPR is probably, I mean, apart from, as you said, being depicted as a bad thing in Hollywood movies and stuff, but it is probably something that would have, I mean, probably it is something that will have a strong impact on humans, on the way we are, literally on what our genome is. So it is important that it is depicted and described in the proper manner, so then people will not be afraid to use it. I will not be afraid to use product that use this technology and can be used in the correct way. You mentioned like the idea of using it for engineering food, because we are doing this to improve the overall health of humans. And so it is important, and we have this opportunity now because it's still quite a young science overall.
And people actually care about it. I mean, these
Yeah,
things, if you explain them properly, or if you help them see what is the value of these technologies. I mean, my favorite example is that during the during the Corona years, when the vaccine started to come out, so many of my friends, they were talking about like, yeah, and I read this study, and this vaccine produces more antibodies than this other one, and this immune response is like that, and so on. So, at that moment, we all cared a lot about COVID.
yeah,
And people were ingesting an insane amount of information to try to find the best vaccine for them and the risks and side effects. So I mean,
absolutely.
and it's also an example of how, in the face of crisis, we can actually innovate, because some of these vaccines, they were produced and tested and evaluated and proved safe in a record time.
Correct.
um,
Yeah,
and some of these, you could consider them, proper GMOs. I mean, they are hybrids of different pieces of DNA from different organisms. So
absolutely.
yeah.
And I mean, it's a shame that we always have to face a crisis in order to advance, that we cannot, do it without the need for, like, what happened with the COVID and in the last years. And it's important for us that we keep the innovation moving forward, even when things are going, let's say, well. Of course, for general public, it becomes more important. So then they become more open to understand, they become more interested, and then you could see already, the interest is going away, and it seems that we want to do, to play as God or whatever. And this is, I mean, that's another problem. I mean, we have to keep the interest alive, because these are problems that we're still facing. I mean, the environmental crisis is still there,
Yeah.
the health crisis is still there. I mean, we haven't finished.
No, we haven't finished. We do live in a period of time with a lot of challenges, and I think that as scientists, we should be able to help people see the connection with our technology and the potential to solve those challenges, I think. Yeah, so there is something to work on. um,
Amazing, very, very interesting. No, I mean, this is a nice message as well. So, I mean, we talk about a lot of things. Of course, CRISPR was just the main topic, and I think we gave an idea of what are our opinion, your opinion, and how this is done, and how it can be used in many applications beyond, genetic editing, and how it can be used for many, many things that are good for humans and in general population. Uh, I think that, and I always like to conclude the episodes like this. No, I think this is something that would interest a lot the new generation of scientists, people that are now entering synthetic biology. Or you already mentioned that you sometimes talk with people with bachelor degree or master degree that they have just concluded. And what would you say to them? I mean, what would you uh, I mean, if you have to think about something inspiring for them, or someone that would be interested in entering this world now, or is now looking for, I don't know, a change of career, or they want to look to start using these methods, or entering the world of gene editing in general, what would you say for as a closing message?
yeah. So for the CRISPR part, I would say like look beyond gene editing. CRISPR has so much potential for other things beyond only gene editing. I think that in the next years, we're just going to see now new technologies derived from CRISPR that maybe have nothing to do with that part. So yeah, I mean, for the CRISPR message, for the early career scientist, yeah, I mean, I would say like, yeah, find what makes you tick, what makes you passionate, and make sure also that that you develop it in a space that supports that. I was very lucky to have a really fantastic mentor during my PhD, Morten Sommer. I did my PhD in the Technical University of Denmark. And I mean, everything that that I know about innovation and entrepreneurship and like how to translate academic research into a company is from him. So make sure that when you start your scientific career, you find a lab that not only has nature papers or like science papers, but that also has good leadership.
Nice, good point. Very good point. Actually, this is something that is never told enough because, as you said, many people look at the impact factor and how many papers they can get in their PhD, and then they are, as you said, left out from the real world. The real world is also that, no, how we translate that into real benefit for the population.
Yep.
Okay, Ruben. I mean, I think we wrap up. I thank you again because you've been a
Yep.
fantastic guest. I hope you enjoyed being here with us.
Yeah,
And
indeed.
for everyone listening, thank you and speak soon.
Yeah. Thank you, Lesio.
That's a load of science.